Okay, so welcome back to this section. My name is Maria Klitschnik and I'm from the University of Salzburg, and I'm working in a group that focuses on the tissue immune regulation. Specifically, we focus on the role of T cells within the human skin immune system. And during my PhD, I developed a mouse model that would allow to study this human skin immune system in vivo. Before we start, a short introduction. So as we've heard now, knew before, the skin is the outermost barrier of the body to the environment, so therefore it encounters constant toxins, pathogens, and trauma. But it's much more than just a passive physical barrier, but it is a very complex immunological organ. And as such a complex immunological organ, it harbors a variety of immune cells. And these immune cells, they do not only interact with each other, but also with the skin cells, so keratinocytes and fibroblasts. This interaction is crucial to maintain tissue homeostasis, support tissue regeneration, while at the same time ensure adequate response to harmful threats such as pathogens. Additionally, we've heard that besides these immune cells, the skin is covered with commensal bacteria or fungi, and these commensals can also beneficially contribute the outcome of immune responses within the skin. So as already mentioned, in our group we focus on the role of T cells within the skin, and a lot of knowledge regarding T cell function in the human immune system has been obtained from murine studies. But when you look at mouse skin and human skin, you can already see that they differ in their structure and also in their cellular composition. So therefore, direct translation of findings from the mouse system to the human system is sometimes a bit difficult. So therefore, there is a need to develop an in vivo mouse model that would allow to study this tissue immune system in vivo. So why are we so interested in T cells in the skin? When you look at the skin, you can see that it harbors 20 billion T cells, which is double the number of T cells found within the whole blood circulation. Additionally, it has been shown that the skin-resident T cells are able to lead to a full-blown immune response, even when we cut the supply of fresh T cells from the blood. So these huge numbers and their distinct function leads us, like many other skin researchers, to the overarching question of what are the factors that control these T cell numbers and function within human skin. So therefore, I set out to develop this humanized mouse model to study human skin in vivo. And when I started to establish or to think about the model, I had some aims in mind that I wanted to fulfill with the model. First of all, we wanted to be able to follow human T cell recruitment to the human skin tissue in steady state, so meaning in absence of any inflammation. Furthermore, we wanted to see if we can elicit and also follow antigen-specific T cell responses in this human skin tissue. And last but not least, we wanted to study the contribution of the skin tissue itself and the contribution of the factors that are secreted by the skin to T cell function and maintenance within the skin. But importantly, the overall aim that we had in mind was to provide the scientific community with a tool that would allow to study the cutaneous immune system in vivo. So hopefully it will be of importance to a lot of you guys. So how does our model look like? How is it set up? On the one side, we have our healthy human donor, and out of this healthy human donor, we isolate keratinocytes and fibroblasts. We expand these cells in vitro, and then we transplant these cells on immunodeficient mice. We use a strain that has no functional immune system of themselves. And for the generation of this engineered skin, we use a specific grafting method that I will talk to you about in a minute. So therefore, we generate simplified skin. Additionally, or in parallel, we isolate peripheral blood mononuclear cells out of the same patient and adoptively transfer these cells into our immunodeficient mouse. So by doing so, we now created a vessel that would allow us to study the T cell infiltration and function within human skin in this immunodeficient mouse. So all the immune cells that we will look at will be of human origin. So why do we bother generating this engineered skin, and why don't we just use full thickness human skin? So there are a bunch of humanized mouse models out there that use full-thickness human skin that was, for example, obtained via bunch biopsies, and just transplant this full-thickness skin onto immunodeficient mice, and then they transfer, for example, human leukocytes into this mouse. However, when you think back to what we heard in the past few days, you know that the human skin harbors a variety of immune cells. So each and every effect that you can detect in the skin will be influenced by a variety of different signals that are obtained from different cell types. So ideally, we thought we start with an empty skin that consists of keratinocytes and fibroblasts only. Thereby, we have a blank slate that would allow us to specifically study the contribution of any cell type that we transfer via IV injection into our mouse, and then study their function within the human skin tissue in vivo, and for example, their response towards trauma, the contribution in tissue regeneration, or even microbial control, commensal microbe control growth. So, how does this engineered skin generation look like in real life? After the expansion of the cells, we use these little silicone grafting chambers. I brought one, but you can also see it here. And it's basically a little head that we place through a small incision on the back of the mice. This head stays in place, and then we just pipette a mix of keratinocytes and fibroblasts into this chamber. The chamber stays on for a week, and after a week it gets removed, and we can see some gooish tissue is formed. And then the wound undergoes stages of wound healing, including wound contraction and scab formation. And after around 4 weeks, we are left with a small pink hairless patch of skin. We performed H&E staining, and we could see that the engineered skin shows features of human skin architecture, such as a multilayered epidermis and the formation of the stratum corneum. Also, the engineered skin is able to produce human structural proteins like type 7 collagen that's produced in the basal membrane zone. So therefore, we suppose that it's correctly assembled and also of human origin. So knowing that we're able to generate this empty skin in vivo, we wanted to know whether we can follow T-cell recruitment in absence of inflammation, so after wound healing, or is complete in this model. Therefore, again, we generated this skin, and then we adoptively transferred human PBMC, and after around 30 days, we looked into the infiltration of cells in the engineered skin. And we wanted to see if we can really follow this tissue-specific recruitment. So when we looked into the splenic and the, now it's working. So when we looked into the splenic and the skin engraftment of human leukocytes, which are defined by expression of the marker CD45, we can see that the engraftment levels are equally well in spleen and engineered skin. And very interestingly, but it has been shown before, the majority of these leukocytes are human CD3 positive T cells. So additionally, we looked at which subsets would engraft or make up this T cell population. We found that gamma delta T cells, as well as CD4-8 and CD4 positive T cells engraft within the model. And importantly, these T cells maintained their functional cytokine profile when we restimulated them ex vivo. So now very interesting was to see whether the T cells that go to the engineered skin, that if they go preferentially to the human skin or if they just go to any skin tissue. So we checked the mouse skin adjacent to the engineered skin for T cell numbers, and we could find that the human T cells prefer to engraft in the engineered skin, thereby already indicating that the human skin tissue may contribute to T cell maintenance in the skin. Okay, so with that, we concluded that we are able to successfully reconstitute the skin T cell compartment using our model. But now we wanted to know whether with the model we will be able to follow antigen-specific responses in vivo, again in steady state. So not in an inflammatory setting, but in steady state. So before we answer that question, I will take you one step back and remind you that human skin does not only consist of keratinocytes, fibroblasts, and T cells, as our skin does now, but also, especially for antigen-specific T cell activation, antigen-presenting cells are very important. And in the skin, we have already heard about it, there are two types, Langerhans cells and dermal dendritic cells. And specifically, dermal dendritic cells are really important to activate resident T cells locally. So, in order to answer the question if we can follow antigen-specific responses in vivo, again, we had the same setup where we generated engineered skin and adaptively transferred human PBMC. And now we started to challenge these skin T cells by intradermal injection of antigen-presenting cells that were either just activated with LPS or loaded with an antigen. And for this study, we chose Candida albicans as a model antigen, as it's a common fungus of the human skin. It's a commensal. And every one of us should have a memory pool, a memory T cell pool towards Candida albicans. So we looked again on the activation and infiltration of human leukocytes and T cells in the engineered skin as well as in the spleen. And we found that upon injection of Candida albicans loaded monocyte-derived dendritic cells, we have a slight increase in human leukocytes. And when we look closer into the T cells, so the CD4 positive T cells in response to Candida albicans, they showed increased proliferation and significantly increased activation compared to CD4 T cells that just encountered LPS-activated monocyte-derived dendritic cells in vivo. So with that, we know that with the model, we are able to follow not only T cell recruitment to non-inflamed skin, but also the antigen-specific activation in this tissue. But as many of you that work with primary human samples know, that access to matched tissue, so all of this was matched skin and PBMC, and all of you that work with these tissues know how hard it is sometimes to get matched tissue. So what we wanted to know is if with the model we will also be able to follow antigen-specific T cell activation in a mismatched setting. So what I did was I used engineered skin from one donor and engineered skin from another donor, and then both of these mice received PBMC and dendritic cells, all derived from this same donor A, creating a mismatched setting here. And again, we injected the antigen and looked for activation and cytokine production in response to the antigen, but also a potential response to the allogenic keratinocytes and fibroblasts. And what we found was that activation of cells, so HLA-DR expression in CD4 positive T cells, was significantly increased in response to the Candida albicans antigen, irrespective of whether the tissue was matched or mismatched. Also, cytokine production was specifically increased in response to Candida albicans. So IL-17 production, there was only a tendency to, visible, but for TNF-alpha we can really detect an antigen-specific secretion. And importantly, when we looked at markers that would indicate an allogenic response, we couldn't see any specific increase of interferon gamma-producing cells in the allogenic setting. Neither could we detect CD8 T cell infiltration of the allogenic tissue. So therefore, there is no indication of an ongoing alloreactive response, at least at the time point that we looked at, which was again 28 days after adoptive transfer of the human PBMC. So we think that the model also provides a tool, even if we don't have access to matched tissues, but we are still able to study T cell responses in steady state. Okay, so last but not least, as we all know, the skin itself produces a lot of factors, and we tried to find out which factors may contribute to this specific T cell recruitment and the support of the function. So what we did was we isolated engineered skin grafts and human skin and performed a Luminex analysis that is more or less like 45 ELISA at a time, and looks at soluble factors that are secreted within the tissue. And what we found was that within the engineered skin, as well as the human skin tissue in comparison, factors that are important for immune cell recruitment are secreted at equal levels, and very importantly, cytokines like IL-7 and IL-15 that play a crucial role in T cell function and maintenance are also secreted at equal levels in both tissues. So we think that the engineered skin that we produce really provides a similar milieu as human skin tissue does. supporting cutaneous immune responses in vivo. Last but not least, we wanted to see if the antigen-specific response really depends on these factors or if it's just an antigen-presenting cell T-cell interaction. So what we did was we used NSG mice that only received PBMC but no engineered skin. And as a comparison, the usual setup where we used engineered skin. And again, both of these mice received human leukocytes and Candida albicans injection. And what we found was surprising or interesting. So in the engineered skin, we have a normal high T-cell number as we usually find when Candida is injected. But in the mouse skin, again, as before, numbers of T-cells are very low. But this time, the injection of Candida albicans could not lead to an increase of T-cell numbers in vivo in the skin. So indicating that it's not just the mere reactivation of T-cells by antigen-presenting cells, but the contribution of the tissue as well. So, yeah. And with that, we think, knowing that the skin tissue contributes to immune function, we think that there is a potential tool to study signaling pathways that are involved in human tissue hemostasis. So with that, I'm already at my summary. So we can say that by transplantation of keratinocytes and fibroblasts only, we can in vivo generate engineered skin that resembles human skin. However, it's devoid of any resident leukocytes that would potentially convolute the effects that we are detecting. And upon adoptive transfer of human leukocytes, we are able to reconstitute our skin with the cell subset of our interest and then study the isolate effect of these T-cell subsets on immune responses or on the response against trauma or the regulation of microbial growth on the skin. So upon injection of human PBMCs, they stably engraft within the spleen and they preferentially infiltrate the engineered human skin but not the murine skin. And upon injection of antigen-presenting cells that are loaded with the microbial antigen, we detect local proliferation as well as activation and cytokine production. And also this response is further supported by the human skin tissue. So with that, we think that the model is suitable to study T-cell recruitment and memory formation in the human skin in vivo. Also, we think it's a useful tool to study antigen-specific processes in human skin and to isolate or to assess the impact of skin-derived factors on healthy human skin immune system. Ongoing studies or successful studies where we used the model was that we recently identified a very particular memory T-cell population in human skin, and with the model we could show that this human T-cell population that we found in the blood can generate resident memory T-cells at distant skin sites. And this T-cell population has a potential involvement in tissue regeneration and wound healing. Furthermore, we can study the generation of resident memory-like T-cells from a circulating pool of Candida albicans specific cells. And a colleague of mine is currently using the model to assess the therapeutic potential of a specific subset of cells in a skin disease setting. So taken together, we think that I created a potential tool that can be useful to a broad variety of scientific fields. We can specifically manipulate the keratinocytes, for example, using CRISPR-Cas9, knocking out specific subsets important for recruitment or even for skin differentiation. And by the blockade of specific cytokines, such as IL-7, we could study the impact of these cytokines on a normal immune system. And last but not least, I think a lot of therapeutical components or the safety of therapeutical components could be tested using the model. So with that, I want to close. I want to thank, first of all, the organizers and the scientific committee, and my supervisor, Iris Gratz at the University of Salzburg, and my external supervisor, Daniel Campbell at the BRI in Seattle. And yeah, thank you very much, and I'm looking forward to your questions.
So many questions.
Thank you very much for your very nice presentation. I think that you can use also your model to study cutaneous T-cell lymphoma like Sézary syndrome or mycosis fungoides, and I think it could be useful for that too.
Yeah.
The only question is about your engineered skin, because I'm afraid you can have a mixture of cells, even if it looks like a human skin, but you can have a mixture of mice keratinocyte and human keratinocyte. So I'm wondering if you have tested using different method, or if you don't have a mixture of cells there.
So we didn't perform like a flow analysis, for example, of the engineered skin where we really sorted out the human versus the mouse keratinocytes or fibroblasts. We did some histological assessment of cytokeratin 5 and 6, which shows the differentiation status of the epidermis, for example. But no, you're right, we didn't really confirm that it's a pure human tissue. But the fact that the milieu that is developed within the model, so the chemokines, what I showed you is just chemokines and cytokines, just a few of them, but also growth factors or angiogenesis factors or neuronal growth factors are secreted at equal levels. And I think this does not really prove one-to-one that it's all human, but it supports at least the idea of that we generated the human milieu. But I agree, I think there should be more characterization.
I agree, but sometimes you can have mosaicism with something like 5% of different cells, and to be sure of your system, I give you the advice to check that.
Yeah, thank you.
Thank you.
This is very interesting study.
Thank you.
So after PBMC transplant, you detect T cells in the skin, but you didn't see any dendritic cells in the skin?
No. So the engraftment of antigen-presenting cells within the model, so just in the spleen, it has been shown before that it's very poor. So this is due to the lack of human GM-CSF and SCSF and IL-3 that's produced in that model. But when we looked in engineered skin, also we couldn't find antigen-presenting. A very, very small proportion, but not a stable population. That's why we decided to reconstitute this population externally.
Good.
Meaning that it's not present, or you cannot...
I think that they do not survive. So when we adoptively transfer the monocytes, or maybe because we have to confirm that, but the blood vessels in the engineered skin are murine blood vessels. And I think that the crosstalk between the monocytes and the murine blood vessels does not, so the human monocytes and the murine blood vessels is not that good. So maybe they cannot exit the blood.
And you could also nicely demonstrate that Candida-specific T-cell immune response are induced after injection.
After injection of heat-killed Candida.
Yeah. So do you think this induction is from naive T-cells or memory T-cells?
I think I know it's memory T-cells, because naive T-cells, we looked for CD45RA expression, which is a marker of naive T-cells. And within the model, so it's one caveat of humanized mouse models that you have the mouse environment. So all the human cells that you put in, they will get activated by the xenogenic environment. So therefore, it's a memory response, so we don't really have naive cells. And interestingly, what we could also see was that from this circulating pool of memory T cells, there was a small proportion that developed resident memory markers like CD69 and CD103. So we could follow this establishment of memory at naive skin sites, so without infection.
I see. So you have never transferred only naive T cells into the...
No, no, we didn't, but people did before, and they are turning non-naive, so they downregulate CD45RA. I think after already 14 days, there are almost no naive T cells left.
Yeah, that's very interesting.
Thank you very much. My concern is about the longevity of those human grafts, because there is a lot of contraction in mice, And how long can you maintain this human skin after grafting?
So after grafting, the size of the graft initially, when we place the cells, is about, I think that's 1 centimeter in diameter. And it always depends on the individual mouse how big the graft will turn out. So usually it's around 0.5 centimeter. So it shrinks a lot already. But after that, it's stable in size until, so we checked for 1 year. It's maintained for 1 year. It's not rejected. It's not removed. So I can just say, at least for 1 year, it's quite stable also in size. Although, again, we don't know if murine cells grow in from the sides during that time, as we never really looked at each cell individually.
This is certainly important to check, but it's not likely that there would be chimerism within the graft. We performed before the grafts on the nude mice with human tissue, and there was no chimerism. There was just outgrowth of murine cells at the periphery of the grafts, but not really the mixing of the keratinocytes within it.
Thank you.
Next one.
Do you think that melanocytes can be easily introduced in your system so that some skin disorders such as vitiligo can be studied?
So we are just about to start that. So a new colleague started in the lab who will take over now that I'm almost done with my PhD, and he worked in the vitiligo field, and he will try to seed melanocytes into the skin tissue that we generate and look whether they are really functional and if we can reflect an autoimmune disease also in vivo in the skin. Yeah, but
Okay,
it's very interesting.
I have 2 questions.
Where?
Me?
Sorry.
The first one, what about T-reg in your model?
Very good question. They do engraft. So I might even have a slide to that. So, yes. So they're maintained in the spleen. So these are CD4 positive T cells in the spleen, and also in the engineered skin, although our N is still a little low, but yeah, we have to increase that, of course. But it looks like as if they're stably engrafting. We didn't test their functionality in terms if they are still suppressive in vivo.
Yes.
So that's another interesting.
And the second one is, so you mentioned that you can use it, the model, to analyze microbiota recolonization or something like that. Did you already test on or not?
No, we didn't, but this idea is derived out of the data I showed you in the end. So when we looked at the factors that are secreted, what I found was that the IL-1 signaling in the engineered skin, so everything is quite reflecting the human skin, only IL-1 signaling is impaired in the engineered skin. And in my head, it's because we don't have a living microbiota on this engineered skin, because it has been shown in germ-free mice that IL-1 signaling is reduced in absence of microbiota. So it would be interesting to see if we reconstitute the human microbiota on our engineered skin to see if we can rescue this IL-1 signaling, for example. Also, we study a population that's involved in tissue regeneration wound healing, and it might be that microbiota also influences the recruitment of these cells, but it's still all in the beginning.
And the second one is, so classically it's mentioned that there is too many gamma delta T cell in mice skin and not really a lot in human skin. How could you manage this ambivalence in your model? So relationship, that's the fact that the ratio between mice and human in terms of gamma delta T cells present in the tissue. Did you check this point or it's not?
You mean if I check the mouse skin for gamma delta T cells?
Yes. And the relationship between your graft and the rest of the skin?
So T cells in the mouse skin, although they are really low in grafting, so not enough survival factors, but they do also reflect more or less what we find in human skin. So gamma delta T cells, they do not engraft better in mouse skin than in our engineered skin, so no difference. might be due to the crosstalk of mouse IL-15 to gamma delta T cells. I think mouse IL-15 does not communicate to human gamma deltas, if I'm not mistaken. So maybe it's a missing signal for the gamma delta T cells. Yeah.
Syriza? Yes. I see.
I have just a very short question about, again, your engineered skin. The epidermis in humans is very thick, and the one in mice is very thin. So how does it work, at the interface between the murine tissue and your skin?
That's a good question. So we can see where it interferes, so the mouse skin and the human skin. And it seems as if there is a mixture, so the epidermis is getting bigger and bigger. Sometimes we also see some adnexal structures in that area, which I think are still murine structures, but in the center of the engineered skin, we really have the thick epidermis throughout. So, yeah.
So last one. Last question? Christine, no? Okay. Okay, thank you so much for your talk. Next. The next one, Adèle Moreau, welcome. Papillary and reticular fibroblast.
So, hello, my name is Adèle Moreau and I'm a second-year PhD student. And I would like to thank the organizing committee for allowing me to present my work today that deals with the angiogenic properties of papillary and reticular fibroblasts in vitro. I'm carrying out this work under the supervision of Laurent Muller at the Collège de France, of Florence Ruggiero at the Institute of Functional Genomics at Lyon, and of Brigitte Kloss and Civil Board at SciLab at Brive-la-Gaillarde. So what is well known about the dermis? It's that it can be subdivided in two main compartments, a superficial one called the papillary dermis, separated from a deeper one called the reticular dermis by a vascular barrier. It is well known that they show different extracellular matrix or organization and composition, and it's also well known that they have cellular differences, as the papillary dermis is more dense in cell compared to the reticular one, and as well fibroblast identity between the papillary and the reticular dermis differs. However, what is not as well known is that there are differences in the vascularization between these two compartments. Skin microvasculature is very essential for skin homeostasis. This includes thermoregulation, immune cell trafficking, oxygen and nutrient supply. It has a very specific architecture that you have here represented. We have a very dense subpapillary plexus that then goes under many capillaries in the papillary dermis that connects a subcutaneous plexus via a network of arterioles and venules. As a result, the papillary dermis is much more vascularized than the reticular ones, and the nature and the morphology of the vessels of these two compartments differ. However, to this day, there is no clear explanation for this difference in vascularization. This is why the aim of this project is to understand what factors can explain the difference in vascularization between the papillary and the reticular dermis. Can papillary and reticular fibroblasts themselves generate a microenvironment that would regulate cutaneous blood morphogenesis? Or are these differences due to a skin-native microenvironment regulation, or even can they both mechanisms explain these differences? To answer these questions, we asked ourselves other questions, including, can papillary and reticular fibroblasts on their own generate a microenvironment in vitro that can sustain the formation of vasculature with the same characteristics that we see in native skin? To answer all of these questions, we decided to carry out a differential analysis of papillary and reticular fibroblasts. We first took a look at their angiogenic properties by using two angiogenesis models. The first one looking at the paracrine secreted factors for both types of fibroblasts in 3D, and the second one looking at the effect of the fibroblast microenvironment itself on capillary formation in a co-culture angiogenesis model. In parallel, we carried out a differential analysis of gene expression and took a look at the angiogenic profile and matrisome of both papillary and reticular in vitro microenvironments. What we know about papillary and reticular fibroblasts is that there are distinct skin fibroblast populations. It was demonstrated using lineage experiments that they are coming from common fibroblast progenitors that then specify during development. And various in vitro and in vivo studies demonstrated that they play different roles in skin physiology, including aging, wound healing, epidermis differentiation, and cancer. In this study, we used the papillary and reticular fibroblasts isolated from abdominal or breast surgery from 3 healthy female patients aged between 20 and 50 years old. What we did is that we mechanically separated the reticular from the papillary compartments, and we discarded the epidermis. And then after all, we put in culture the explants from the papillary and the reticular compartments. We then defined the papillary and reticular cell cultures based on the morphology and the proliferation rate of the different cell cultures. And so we defined the papillary fibroblast cell culture as a culture with elongated cell, as you can see here, with a high proliferation rate. And on the contrary, we characterized the reticular cellular culture based on the presence of a lot of spread stellar cells with low proliferation rates. In this study, we used both 3 pairs of fibroblasts that we cultivate in over-confluent culture, and we stimulated the deposition of extracellular matrix, and I will refer at this culture as cell sheets in this presentation. So, using these cell cultures, we took a look at the angiogenic properties of these fibroblasts using these 2 models. The first model allows us to study the contribution of fibroblasts' secreted factors. It's a 3D angiogenesis model. We are in a fibrid gel, and we use fibroblast conditioned media. And the formation of capillaries in this model is similar to spreading angiogenesis. In the second model, we use a model where we study the contribution of the fibroblast microenvironment themselves. So we let the fibroblasts do their own extracellular matrix, and then we see how it affects capillary formation. And this model is mainly recapitulating characteristics of vasculogenesis. Looking at the first model, so we first wanted to take a look and to see if papillary and reticular fibroblasts secrete distinct factors that could differentially regulate capillary formation in 3D. To do so, we use dextran beads that are coated with gelatin, and we cover them with primary endothelial cells from umbilical cords called UVEC. We then have this nice covered bead that we include in a fibrin gel in the presence or absence of vascular endothelial growth factor. We then add on top of this hydrogel papillary or reticular conditioned medium. And after 4 days, as you can see in this 3D representation, the endothelial cells sprout from the bead and they form in 3D capillaries. This model recapitulates sprouting angiogenesis, the main characteristics, including tip cell and stalk cell specification, the deposition of a basement membrane that you can see here by deposition of collagen type 4, and the presence of a lumen that you can see here on this image of scanning electron microscopy. So you have here the bead, here the capillaries, and the lumen that you distinguish inside. So using this model, what we observed is that the papillary fibroblasts are able to secrete factors that could sustain the formation of a lot of branched capillaries independent of VEGF stimulation. That means that papillary fibroblasts have a really high angiogenic potential. On the contrary, the reticular fibroblasts couldn't secreted factors, had a low angiogenic potential, but were still able to sustain the formation of capillaries in response to VEGF. Looking at the involvement of the fibroblast microenvironment now. So in this co-culture angiogenesis model, what we do is that I take the papillary and reticular fibroblasts that I cultivate as cell sheets, as I mentioned before, and they have the time to deposit a nice extracellular matrix that you can see here on this second harmonic generation image that I did with 2-photon microscopy. So then I sit on top of these layers UVEX, and I allow them to grow and to organize into capillaries. And after a week, we can observe a capillary formation using the endothelial marker CD31. You can see that we have capillary formation on top of fibroblast layers, and in this model we have as well capillaries that are mature, as indicated by deposition of type 4 collagen. I then quantified the vascular network that was done using this model, using a macro that was developed at the laboratory. So to do so, we imaged the whole cell culture well, and then we choose a region of interest. We then generate a binary mask from which we deduce the percentage of area covered by vessel that we refer as vascular area. We then generate an image map from which we can calculate the mean diameter of each vessel. And then from all of this, we generate a skeleton, and by undergoing a skeleton analysis, we can look at vessel length and number of branch points. Using this model, so in the absence of VEGF, there is no difference in capillary formation between papillary and reticular fibroblasts. But you can still notice that both types of fibroblasts in this model can induce the formation of capillaries. What was interesting, though, is that in these conditions, the microenvironment generated by reticular fibroblasts is able to induce the formation of vessels of larger diameter, as verified by quantification. When stimulating with VEGF, we could clearly see that the papillary microenvironment was able to induce the formation of a much more denser vascular network with a lot more branch points, as verified by quantification as well, because we have a higher vascular area, number of branches much more important, and a vessel length as well much more important. So by using these two angiogenesis models, we first demonstrated that papillary fibroblasts have a higher angiogenic potential compared to the reticular ones, and that both papillary and reticular microenvironments in vitro can induce the formation of vessels that reproduce main characteristics of blood morphogenesis observed in vivo. So going further, we asked ourselves, what factors can explain these differences? And to have a more whole approach, what I decided to do is to do a differential analysis of gene expression. So past studies, there are a lot of studies already published on transcriptomy studies about papillary and reticular fibroblast gene expression. And most of them, they demonstrated that papillary and reticular fibroblasts, they differentially express different family of genes involved with different function. And they also identified identity markers, including extracellular matrix components. And recently, there was also a demonstration that there is a switch of identity related to aging. However, these studies were done on non-confluent cells at early passage, and most of them used a microarray approach. This is why, to answer our questions, we decided to culture the papillary and reticular fibroblasts as cell sheets, and then to, as in angiogenesis models, and then to perform an RNA sequencing using the Illumina technology synthesis, sequencing by synthesis, in order to increase the depth of our analysis. So the first thing that we wanted to do is to make sure that in this conditional culture, we still have two different populations of cells that keep their identity. To verify that, we did a principal component analysis, and we observed that the samples segregate into main groups corresponding to papillary and reticular fibroblasts. We then defined the differentially expressed genes as the genes having a ratio superior, fold change superior to 1.5, with a p value that was significant. And we identified 102 differentially expressed papillary genes and 1,244 reticular genes, with more than 50,000 that stayed equally regulated. And among these differentially regulated genes, we found markers that was previously described, such as podoplanine for papillary fibroblast, collagen 7, and for reticular fibroblast, collagen type 11, and FGF7. So we had two different cell sheets with specific identity. Then we wanted to take a look at what biological process that could distinguish these two cell sheets. And to do so, I performed the bioinformatic analysis, and I gathered all the differentially regulated genes that are entered in a David bioinformatic resource. And then I extracted all the Go terms that were the mainly enriched in this condition of culture. And what I observed is that you have here the 20 most enriched Go terms. It's that among the 5 most enriched ones, we found 2 related to extracellular matrix and matrix cells adhesion that are here in green, and also 2 that are related to angiogenesis and its regulation. So it means that in this condition of culture, we have papillary and reticular fibroblasts that are distinguished by angiogenesis and microenvironments. So then we took a deeper look at it. First, taking a look at their angiogenic profile. To do so, I listed all the genes that were differentially regulated for each subtype of fibroblast, and I compared it to the list of the vascular development Go term. Then realized an expression heat map and carried out a hierarchical clustering. You have here a representation of this study. And what I observed is that spontaneously, the samples clustered in 2 main groups corresponding to the papillary and reticular fibroblast samples, indicating that we have a specific signature for angiogenic properties. Then to go further, we asked ourselves, okay, we have difference in genes, but are these genes pro or anti-angiogenic factors? So then I extracted from these genes that I referred as effector of angiogenesis. So effector of angiogenesis, I define them as genes coding for proteins that are able to interact directly with endothelial cells, including secreted factors, ECM components, as well as membrane and core proteins. And to this day, I found almost only pro-angiogenic effector genes expressed in the papillary fibroblast, while I observed a more subtle balance between anti-angiogenic genes and context-dependent genes. So these differences could explain partially the difference that I observed in the angiogenesis model in vitro. And interestingly, I found expression of smooth muscle cell differentiation markers within the reticular fibroblasts, markers that are expressed in some vascular smooth muscle cell associated cells, and that are really important to understand, that could explain as well differential interaction between the endothelial cells and the reticular fibroblasts. To partially verify this with protein, I did an antibody array using conditioned media for papillary and reticular fibroblasts. It's a commercial antibody array with 53 target proteins, and I confirmed the differential expression of some proteins analyzed in RNA sequencing, including collagen type 18 and placental growth factor for papillary fibroblasts, hepatocyte growth factor and chemokine ligand type 2 for reticular fibroblasts. And interestingly, I also observed differential secretion of other non-identified targets, such as VEGF for papillary fibroblasts and interleukin type 8 for reticular fibroblasts. So now we know that with this bioinformatic analysis, we have different expression of pro, anti, and context-dependent markers between the 2 fibroblasts, including angiopoietin type 1, anti-angiogenic regulators in reticular fibroblasts, such as thrombospondin type 1, BMP4, and context-dependent, such as NPCL, like angiopoietin-like 4 protein. As I mentioned, the matrix is as well another differentially regulated process. So then we took a look at this process using the matrisome. So the matrisome is a list of genes that lists all the genes that are involved in the structural composition of the matrix and its regulation. And it's available online and published by Naba et al. And it's subdivided in different categories, such as the secreted factors, regulators, ECM affiliated. And the matrisome itself can be subdivided in between glycoproteins, collagens, and proteoglycans. So in a previous study carried out by my teams, there was a matrisome that was characterized for the freshly isolated papillary and reticular fibroblasts. And in this study, we identified the markers of ECM from these 2 types of fibroblasts, and we also observed that the reticular fibroblasts express more genes related to the matrisome, and especially structural components of this matrix. So taking this as a reference, we then took a look at the matrisome of the cell sheets that we generated here, and we had an increase by 4 times of the number of matrisome-associated genes. And interestingly, this difference was not equal between papillary and reticular fibroblasts. We find 5 times more genes in the reticular fibroblasts an increase, and only 3 times in papillary fibroblasts. And this, we found more ECM-associated genes. And looking at the structural composition, so the core matrisome, we found the same trend, and with more glycoproteins. So now, the same as for the androgenic profile, I took a look at I made the heat map expression of all of these genes, and I'm presenting you for each subcategories the results that I got. So, using hierarchical clustering, what we could observe is that there was a spontaneous, as well, gathering of the samples according to papillary and reticular identity, meaning that in this condition of culture, we have papillary and reticular microenvironments that have specific matrisome signature. We then found as well previous markers that were defined, especially in the P1 study that I here referred in purple. Interestingly, we find twice more non-fibrillar collagens expressed in the papillary fibroblasts. And we found as well 6 times more proteoglycan that were differentially expressed in the reticular fibroblasts. And we enriched the number of genes associated to the elastic fiber assembly in the reticular fibroblasts that are here highlighted in red. We did the same, and I'm still doing the same for the matrisome-associated genes. And to this day, we have very interesting lead. And I notably noticed the presence of genes coding for protease inhibition of in the reticular fibroblast, and notably as well identified actors of immunity in the ECM-affiliated genes. So to summarize what we learned with this study, so using 2 models of angiogenesis, we demonstrated that papillary and reticular fibroblasts have different angiogenic properties. This is due to a higher angiogenic potential in the papillary compared to the reticular one. Plus, by their own in vitro, generating their own microenvironment, they can reproduce some of the features that are characteristic to cutaneous blood morphogenesis. To this day, using RNA sequencing, we here propose that these differences are due to a difference in expression of gene involved in the pro- or anti-angiogenic regulation of angiogenesis, with more genes involved with pro-angiogenic in the papillary versus anti-context-dependent in reticular fibroblasts. We also identified markers of smooth muscle cells that could explain that could be responsible for different interaction between the reticular fibroblasts and the endothelial cells. And we demonstrated that they have a specific matrisome. So what's going next is to understand what functional links can we make between the microenvironments and the regulation of angiogenesis in the papillary and reticular microenvironments. And this is the first step to understand what is the regulation of the formation of vessels within the skin, and it's very important for tissue engineering perspective and even for therapeutic perspective. So I would like to thank you very much for your attention and to thank everybody that contributed to this work at the Collège de France, at the IGFL, and at CILAB. Especially thank for Pauline Joncourt, who did all the biostatistic work. Thank you to the engineers of the platform sequencing at the IGFL de Lyon, and thank you very much for the past and present engineers of the core facility at Paris. And I'll be happy to answer questions.
Thank you. Some few questions, because I have. So just, you mentioned that therapeutic application.
Yes.
Which kind?
So it's future application. What we can think about is what kind of fibroblast for what kind of graft. For example, as we see that the papillary fibroblasts have a much higher angiogenic potential, what we can think of is for a chronic wound. We can preferably do graft with papillary fibroblasts compared to the reticular one. And I think it's also important to emphasize that as the reticular fibroblasts by themselves can also do blood morphogenesis, perhaps in few years we will be able to find applications for reticular fibroblasts themselves in other contexts. To this day there is none, but I think this is going to come as well.
could we consider that so one of the population controls the other one?
Yeah. Well, so this is really interesting, and definitely this is something that we have to look at. In the previous study, in transcriptomic, it was demonstrated that there could be some crosstalk looking at the regulation of some signaling pathway. And I would definitely say that it's more likely that there is a communication between the two subtypes to maintain the blood structure. Particularly because in the skin, it's true that in adult skin there is no angiogenesis except in disease or except in wound healing. So I would say there is kind of a status quo between the two compartments.
And the last point, after that I stopped you. So, and what about neuromediators? So you don't mention CGRP, NPY in the control of all this angiogenesis, because so you don't find anything in your global screen? Or?
So I didn't find differential expression in this screen. And it's important to, I would like to emphasize as well that I only took a look at the differential expression. But it might be that right now I can answer because I don't have in mind the list. But I would not be surprised if there is an equal regulation of these factors between the two types of fibroblasts.
Yes, I think so.
I have a question about your RNA-seq analysis. Did you analyze the data at the gene level or at the transcript level?
Wait, sorry, at the gene level?
Gene level or at the transcript level? So do you think it could be of interest to analyze the data at the transcript level and not only at the gene level?
Do you mean the level of transcripts?
Yeah.
The number of reads that I will find for each?
Yeah, you have the reads, but you can summarize all the reads for one gene, or you can identify the different transcript, potentially differentially splice or variant of transcript for each gene. So I would like to know if you could be of interest of doing this sort of analysis.
So here is level of expression of genes, but not spliced variants. This is the question, right? So I didn't do it here. Yeah,
so it depends the way of sequencing, if it is per hand or single hands. But if it is per hand sequencing, you can have the gene variants in your data.
I don't have the gene variants here.
Yeah. Okay, thank you.
But it's definitely something that we have to look at. That would be interesting.
So just a comment before
yes.
I think that so we have a session this morning on microbiome control, and so people work on this kind of approach to understand neovascularization of the skin, but nobody's use any kind of strain extract or strain mediator coming from the local commensal population to try to know if yes or not there is an increase of vascularization that this kind of strain can provide something to the skin, even in deeper, because so Rich Gallo demonstrated a few years ago that we could find some bacterial strain until the basal layer. I think that maybe you could have a look on this field. So.
And it's particularly interesting because we found some immunity markers that are well known in papillary fibroblasts, right? So, including antigen recognizing molecules. That definitely would be really interesting to look at.
Maybe you could exchange with the guy this morning, coming from Lund.
Yes.
Okay, thank you.
Thank you very much.
The last one. So, Sindécan, Marine Montmasson, correct?
Yes.
Okay. So, just explain something.
Okay, so good morning. My name is Marine Montmasson. I am a PhD student in third year in the cell microenvironment crosstalk and tissue repair lab headed by Patricia Roussel at the Laboratoire de Biologie Tissulaire et Ingénierie Thérapeutique of Lyon. I really want to thank the scientific committee for selecting my abstract, and I'm really glad to present you our work entitled Sandecon-1 drives MMP9 expression during keratinocyte migration within epithelial podosome-like structures. So during skin repair, the reepithelialization step is essential to restore the skin barrier functions. The cells localized at the bond edges are subjected to morphological modifications and acquire a migratory phenotype. They develop specific adhesion structures and acquire cytoskeletal rearrangements exercising cellular motility. Frontal migratory cells apply an attractive force on neighboring cells, leading in collective migrations where movements are coordinated. This process leads to a confluent cell layer where keratinocytes begin to divide and differentiate. So these events are complex and depend on the production of growth factors, matrix metalloproteinases, or extracellular matrix proteins on which keratinocytes adhere and migrate. The laminin 332, which is the major adhesion protein of keratinocytes, was shown to be involved in regeneration. So this extracellular matrix protein, shown here, is composed of 3 chains, alpha-3, beta-3, and gamma-2, assembled in a triple helix. This basement membrane component is the first protein deposited by migrating keratinocytes as a precursor form. The cleavage of 2 globular domains called LG45, localized at the C-terminus of its precursor alpha-3 chains, leads to a mature form promoting cell adhesion. In 2003, my lab showed that the interaction that the LG45 domain interacts with the Sandecon-1 receptor. So this transmembrane receptor is a member of the heparan sulfate proteoglycan family. It has a cytoplasmic tail composed of 2 conserved domains C1 and C2 and variable region V, a transmembrane domain and an exodomain supporting 2 types of glycosaminoglycans, the heparan sulfate and the chondroitin sulfate. It interacts with several molecules like growth factors, cytokines, or extracellular matrix proteins like the laminin 332. And in 2007, Patricia Roussel's lab showed that the interaction between the LG45 precursor domains and the heparan sulfate of the Sandecon-1 participates in keratinocyte migration. So during wound healing, keratinocytes express several matrix metalloproteinases, or MMPs, to migrate into the wound bed. Thanks to their enzymatic activity, they perform several functions, like the activation of other MMPs and bioactive molecules, the disruption of cell attachment to the provisional matrix, the cleavage of cell-cell junctions, or the degradation of basement membrane components. During reepithelialization, several MMPs are produced at different levels of the wound. So as you can see, the MMP 3, 19, and 28 are expressed behind the front edge as site of proliferative epithelium, while MMP 1, 9, and 10 are expressed at the tip of the migrating tongue. So MMPs are multi-domain zinc containing endopeptidases, which are involved in tissue remodeling and repair. They are produced as a latent form called zymogen, which requires proteolytic process to be fully active. The MMP9, which is a member of the gelatinous group of MMPs, is also called gelatinase B, and is not expressed by keratinocytes, but is upregulating during skin healing. It cleaves collagen 4 and 7, the elastin and the fibronectin, but also the gelatin, and is regulated by several molecules like TNF-alpha, IL-1 beta, TGF-beta, or the interactions with the substrate and alpha-3 beta-1 integrin. Its absence results in compromised reepithelialization due to the retardation of keratinocyte migration and the reduced clearance of fibrin clots in MMP9 knockout mice. However, its overexpression is associated with chronic wounds due to impaired restoration of the basement membrane. So recent results from my lab have reported that the LG45 domain of the precursor laminin 332 upregulates the MMP9 expression in normal human keratinocytes at both transcriptional and protein levels. So as you can see on the zymograph of the in-gel gelatin zymograph analysis of the supernatant of NHK treated with LG45, we observed an MMP9 expression increased while the MMP2 expression is not affected. An in-gel gelatin zymography consists in the incorporation of a gelatin during the preparation of an acrylamide gel. After electrophoresis and the staining thanks to Comasy blue, the areas digested by the gelatinases MMP2 and 9 appears clearer. So in this experiment, the conditioned media of HT1080 cells were used as a control to verify the molecular weight of the band, because these cells were known to express high levels of both gelatinase 2 and 9. Finally, treating cells with increasing concentration of LG45, as you can see on the zymogram and its quantification, induce a dose response of MMP9 expression activation. So in order to know if Syndecon-1 is involved in LG45-induced MMP9 expression, we perform a siRNA knockdown approach of this receptor. The depletion was confirmed by FACS analysis, and the MMP's expression were evaluated by zymography. So as you can see here, we can observe the increase of the MMP9 expression in our control transfected cells, but this increase disappears in the cells transfected with the specific Syndecon-1 siRNA, suggesting that this receptor is involved in this mechanism. Moreover, we saw that the same effect was observed with the treatment of TNF-alpha and IL-1 beta, which are two pro-migratory cytokines which are known to upregulate the MMP9 expression. So these results indicate that the Syndecon-1 seems to be involved in the regulation of MMP9 expression, whatever the active molecule used is. Consequently, we wanted to understand how this mechanism is performing. So then we analyzed the MMP9 expression in NHK lysates in order to know if the variations were found at both cellular level and in the matrix environment. So our in-gel gelatin zymography revealed that the MMP9 expression recapitulated findings from conditioned media, excepted that the MMP9 expression of the active form was found in much higher amount. So interestingly, we found that both pro and active MMP9 were enhanced with the treatment of LG45, as well as with the treatment with IL-1 beta used here as a control, suggesting that most of the active MMP9 remains in the cellular vicinity. So therefore, we have performed in situ demography of keratinocytes treated or not with LG45 in order to locate the MMP9 activity in our cultures. So thanks to several steps and chemical reagents, we deposited a thin layer of gelatin coupled with cyanin-3 on cover slips, where cells are then seeded on. After treatment and 48 hours of incubations, our cells are then fixed and observed by microscopy. Because the MMP2 and 9 are the only ones able to degrade gelatin, their activity can be observed as black holes in the red fluorescent background due to the proteolysis of the gelatin. Thanks to immunofluorescence, the actin appears in green with the use of phalloidine FITC, and the nuclei in blue thanks to the DABI staining. So, thanks to our in situ gelatin demography, we found areas of digested areas resembling adhesion contacts underneath and around the keratinocyte, probably due to the movement of the cells. Moreover, and interestingly, we found that the number of these black dots increased in LG45 as well as with IL-1 beta treatments. The quantification of the total digested areas in each field observed in our cultures showed that the treatment of LG45 and IL-1 beta increased the total digestion surface in several strains of NHK. So to analyze whether MMP9 is involved in the formation of the digested areas, 2 inhibitors were used. The first one specific against the gelatinases family, so it is an MMP2 and 9 inhibitor, and the second one an inhibitor specific exclusively for MMP9. So first of all, we decided to verify the dose-response inhibition of keratinocyte migration in the scratch assays, because a lack of MMP9 impaired cell migration. So while the wound closure 48 hours after injury is about 60% for the control, you can see that the percentage of wound closure decreased as the concentration of inhibitors increased. Our confocal microscopy analysis revealed that the use of the inhibitors prevented the formation of these black dots observed in our controls. And these results are shown in both cases with cells treated or not with LG45, suggesting that the MMP9 is responsible for these digested areas. We have performed several scRNA experiments recently in the lab that confirmed these results. So thanks to further confocal microscopy analysis, we found 2 types of digested areas in our cultures. So as you can see in the cell periphery that mimic the position of the cell protrusion, and also at the center of the cells when we saw again our previously described black dots. Moreover, the number of these black dots is again enhanced in these pictures with the treatment with LG45. But interestingly, we found several actin-positive staining within digested areas in both cases, suggesting that an adhesion receptors may have been involved in their formation. So because these structures look like podosomes, which are specific adhesion structures associated with matrix degradation, we performed confocal microscopy analysis of one of the major components of the podosome, which is the cortactin. So as you can see, this protein is organized as blue dots located inside the digested areas, like actin before. And at the same time, we stained the Sendecon1, and this receptor appears organized as a ring surrounding the digested areas, but also the blue dots of cortactin, as you can see on these pictures. So this result suggests that there are several cellular processes involving at least Sendecon1, actin and cortactin, which are responsible for MMP9 deposition, most likely in podosome structures. So consequently, we decided to answer 2 questions. Is the MMP9 digestion dependent of Sendecon1? And can the observed cell structures be podosomes? So thanks to scRNA Sendecon1 approach, we decided to answer the first question, which is if the MMP9 digestion is dependent of Sendecon1. So as you can see in our control cells, we found the previously described ring of Sendecon1 surrounding the black dots, but also our dots of actin. Nevertheless, we observed several significant changes in our cultures of cells with, which are transfected with the specific Sendecon1 scRNA, like the loss of the Sendecon1 rings that confirmed the efficiency of our transfections, but also the decrease of the size of the actin dots. Moreover, the quantification of the digested areas revealed that the lack of Sendecon1 drastically decreased the total digestion surface, but also the digestion surface observed per point. So this result suggests that the Sendecon1 is involved in MMP9 expression and activity. So regarding to the second question, which is, can the observed structures be podosomes? We wanted to know if our cellular processes involving actin, cortactin, syndecon-1, and MMP9 activity can be podosome-like structures. So the podosomes are dynamic adhesion microdomains which are located at the ventral plasma membrane of our cells, as you can see in these pictures. It has a characteristic structures with an actin and associated cytoskeletal protein cores, as you can see, and a signal and structural protein organized in rings associated with MMP's activity. So in order to characterize our observed structure, we performed immunostaining of the core proteins, and especially of WASP, alpha-actinin, VASP, and ARP23. And the results are shown here. So as you can see, all the actin regulatory proteins analyzed match the digested areas as blue dots located inside the digested areas and surrounded by the syndecon-1 rings in all our immunostaining. So these results match with the podosome literature. So we are actually performing immunostaining of podosome ring proteins with vinculin antibodies in order to stain the cortex. And these results are actually in process, and we found for the first experiment that this is the case. So we found that the vinculin stained the cortex as expected, like the syndecon-1. So our results revealed that syndecon-1 is a new component of the podosome structures, which is involved in their formation, but also in the control of the MMP9 expression. For example, we can make the hypothesis that a potential partner for the syndecon-1 could be the CD44. Indeed, this hyaluronic acid receptor was shown to bind the cytoplasmic tail of the syndecon-1, but also to interact and activate the MMP9. So taken together, our data demonstrate that syndecon-1 plays a role in MMP9 expression and deposition, but is also a component of MMP9 epithelial podosomes. It is involved in LG45-induced MMP9 expression, and its redistribution at the front edge of migrating keratinocytes may have a role to play in the cleavage and the degradation of extracellular matrix proteins, facilitating their pass through the Frebring clot. So I really want to thank Patricia Roussel, my PhD supervisor, and all the people of my lab, especially Gila Dayan, Cécile Garnier, and Anna Michoupoulou, our collaborator Elise Lambert, and the platforms of our campus, the Platim and Anira. This project and myself were funded by the ANR Skin Healing. And finally, I would like to thank the scientific committee and Greentech for allowing me to present our work and you for your attention.
Question? Two?
So very nice presentation, Marine.
Thank you.
So gelatin is somehow an artificial protein because you will not really find gelatin in the skin. So it's supposed to represent denatured collagen, I think. So what do you think about the status of collagen denaturation in the skin bed and in the skin wounds in general? Do you think there is more denatured collagen in wounds? And is it supposed to represent this physiological or pathophysiological setting?
So I think that we can find a sort of gelatin in the wound because of the activity of the collagenases which degrade the collagen. Actually, we decided to use the gelatin to detect especially our MMP9 activity or MMP2, but it's not really expressed in keratinocytes. But we performed several other experiments on collagen 1, type 1 collagen with our cultures, and we found again the podosome-like structure. So I think that the formation of the podosome are not dependent of the gelatin or collagen in vivo, at least.
Upstairs. Ah.
I start. So, I just would like to ask you whether, when there is this epithelial, the reepithelialization during wound healing, do you have the migration of the basal cells first, and then the basement membrane is made, or the basement membrane is made as soon as the keratinocytes migrate?
There are two mechanisms which are in competition in the literature, because the cells who migrate into the wound bed are basal cells, but people don't really know if there is first the migration and then the proliferation and differentiation of all these mechanisms are affected simultaneously. I don't know if I answer your question or
in terms of basement membrane, because you are talking about the laminin, etc., the interaction.
Yeah.
And if I follow up the question of Catherine saying, is it a physiological substrate when you are using gelatin? Just wondering whether perhaps the keratinocytes are producing the basement membrane to migrate. Otherwise, the system is a little bit biased.
During wound healing, keratinocytes deposited some proteins like the laminin 332 or fibronectin. Concerning the gelatin, I think it's possible that MMPs, thanks to their degradation, can increase the quantity of gelatin, but I'm not really sure that gelatin is really presented as the gelatin I use in the wound healing, in our wounds, in our human wounds. But yes, keratinocytes deposited several proteins simultaneously that they are migration.
Another question? Just before the microphone climb up, so I have two questions. The first one, syndicant 1, okay, it's one of the heparan sulfate proteoglycan, but so did you check as a type of syndican, like 4, or as a type of heparan sulfate proteoglycan in all these fields?
Yeah, not yet. It would be very interestingly, but we don't perform that yet. But I think that it would be very interesting because we have a redistribution of syndicant 4 in our keratinocytes, so can be another study to find if the syndicant 4 is involved in also in MMP9 expression, but we don't study that.
For neurovascularization, like Perlecan, for example?
Yeah.
Same question for the previous candidate. So, last question?
Yeah, on the back here. You mentioned that Syndecan can control the activity of the MMP9. So I was just wondering whether you would believe that it's direct, or whether it could be by being a kind of structural component of the podosome. And so if you limit the podosome formation or fusion or whatever, you would of course have an impact on the activity.
Yeah, so actually we don't know the real mechanisms which involve Syndecan 1 and MMP9 expression. But thanks to several studies, we found that there is a shading, a physiological shading of the ectodomin Syndecan 1, which involved the translocation of the ectodomain shade in the nuclei to express several genes. And so maybe it can be a possible hypothesis concerning the MMP9 expression, which would be direct. And we found also that the IL-1 receptor is involved in the MMP9 expression, but it is a study of 2011 where we found that HSPG is involved in the IL-1R signal, but this HSPG is not detected, is not identified. Maybe it can be an hypothesis that Sendecolon will be a co-receptor of the interleukin-1 receptor.
Okay, thank you. So, for all attendees, please, 2 recommendations. So please go back at 2 p.m. for the last session, and if you can get only 1 hour and 15 minutes for the lunch. First of all, I see the manager of the foundation with your wife. Go quickly to finalize my comments. So the second point, please finalize your vote for the poster session until 1:30. After that, we get all the results with the jury committees, and please enjoy your lunch. Maybe? No? Not yet?
I have only 2 things to say. One, maybe you know and you saw, but you have a USB key on your pen, so you can use it. Maybe you knew before, but it's important to show you. Another thing, so I talk about Cosmetics 360, and we have another support and partner is IFSCC, and we have a quick 3 minutes presentation for the next year in Milano of the IFSCC 2019 in Milano. So if we can launch a movie in the sky.
I am here to announce the 25th IFSCC conference with the challenging title of Cosmetic, Science and Conscience. The event will be held in Milano in 2019 from September the 30th to October the 2nd.
On the conference website, the universe is used as a metaphor for cosmetic science, whose constant challenge is to find rational solutions to problems that are often anything but rational. The supported texts are represented by constellations, each of which offers a valuable set of innovative benefits. These include the adoption of scientific sessions and donation of part of the supporters' contribution to philanthropic organizations.
Italy has already hosted 2 highly successful IFSCC conferences. This 2019 edition in Milano, the capital of beauty, design and fashion, will be an even greater success. So mark the date in your diary and be sure of joining us.
So thank you to SICC, Italian Society of Cosmetic and Chemistry, and to Elio and Stefania. So thank you for your support to Spim, and welcome in Milano for everybody. You will see it's on the sky. Thank you, and good lunch.